Alright, I think we're good. Howdy everybody. I'm just making sure my camera is all set up. Okay, so what we have here is I got my cookie sheet and I've taken the two colors of clay. I've rolled up about 60 to 65 of the white ones. I've got my 4 centromeres, that's the long kind of skinny ones that go in the middle, and then I got the 4 centromeres of the yellow, and then about 60 to 65 of the yellow beads, if you will. I still had some leftover clay after this was done. I suggest making them about this big. And what you can do that's really fast, you just take a little pinch off the ball and then you roll it like this and then you're done. Take a pinch, roll it, you're done. And they don't all have to be the same size. All right, so the first thing we're gonna do is we're gonna construct 4 chromosomes like they're shown on page 9 at the top. And so we're gonna do this. We'll just take the white centromere and then we'll take 10 of our white beads and we'll put them on the top. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'll take 10 beads and I'll put them on the bottom. I'm going to join them all first and then I'll stick them on the bottom. So I got 6, 7, 8, 9, and 10. Okay, so now we've got 1 chromosome. Now, before we go further, let me just explain. The chromosome that I just made represents the chromosome, one of the 23 chromosomes that you will get from your dad. The ones in yellow will be the one of 23 that you get from your mom. So what we can do is we can call this chromosome number 1, and I should have 2 copies of chromosome number 1. And so I'll go ahead and make the second copy, the one that I got from my mom, and I'll put 10 beads on each side. So we got, that's 5, 6, 7, 8, 9, 10. Okay, I'll do that to each side. Sorry if the camera's jiggling. There we go. I go 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 10. All right, and so that's the first chromosome. So remember, we're diploid, so we have 2 copies of each chromosome. 1 copy we get from dad and 1 copy we get from mom. And these chromosomes are homologous, meaning they're gonna have the same sequence and order of genes on them. They're just gonna vary in terms of alleles because your mom has a different genetic history than your dad, but we're all in the same species, and so we all have the same genes on our chromosomes, but the alleles for those genes are oftentimes different. And so what we'll say is, okay, this is the first chromosome out of the 23 pairs of chromosomes that we have. And now we need to make the second chromosome. And so we'll make this one just with 5 beads on each side. So I'll call that chromosome number 1. I'll call this chromosome number 2. And I'll put 5 beads on each side. 1, 2, 3, 4, 5. And so this is a completely different chromosome than the long one. This is a chromosome, let's say the long one has on it genes that code for hair color and eye color. Maybe the short one has genes on it that code for height and body weight or something like that, you know, baseline height and body weight. Obviously the environment has some effect on those. Okay, and then we'll put 5 beads on each side of the copy we got from mom. So 1 copy we got from dad, 1 copy we got from mom. So we have 2 copies of the same chromosome, identical in terms of the sequence of genes that appear on them, but different in terms of the alleles or the variations of those genes. Okay, so I've done that. I've placed the chromosomes in the center of a cookie sheet, and then we're gonna take a piece of yarn or string. We're just gonna kind of wrap it around those. Oops, I don't want that one, I want the long one. All right, so we'll take a piece of yarn or string and we'll wrap it around. And what this simulates is, oh no, I'm losing some of my chromosome. All right. So this is the nucleus of the cell. These are my chromosomes in the cell. I've got chromosome 1, I have 2 copies. One I got from dad, one I got from mom. Chromosome 2, I got 1 copy from dad, 1 copy from mom. And it would be this way for 23 pairs of chromosomes. Okay, 2 full sets of chromosomes. Okay, now we just need the 2 centrioles just outside the nuclear membrane. So I will put those like that. That's my centrioles. And let's see, and then, so now we need to talk. So this is interphase. This is, the hard part about this lab is that, as you know from our existential DNA lecture, is that DNA can exist in a heterochromatin state, which is what it looks like right now, where it's kind of tight and condensed, or it exists in a euchromatin state. Now, parts of the chromosomes can be in euchromatin, while other parts of the same chromosome can be in heterochromatin state. And so doing it with modeling clay is a little bit tricky because there's no way to show they're all in euchromatin, or they always look like they're in heterochromatin in this stage. Okay, so during S phase, which is the synthesis phase of interphase, the chromosomes are going to replicate. Now, let me be very clear about this. This is DNA replication. This is that whole origin of replication, the replication forks separating, the helicase moving and unwinding the 2 strands, the topoisomerase cutting and unwinding and regluing the strands back together. This is DNA polymerase 1, DNA polymerase 3, the RNA primase. All of that is happening right now. So that's happening on these chromosomes. And what we're going to end up with when we're done is we're going to end up with 2 copies of every chromosome. So I need to make another long white one with 10 beads. We'll call them genetic beads on each side. So I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then I'll make one more. Sorry, this cookie sheet is wobbly. Hope I'm not making you guys seasick. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So now I've just duplicated. So I've gone through S phase for this big long chromosome. Try to keep them close together there. Okay, so now I got 2 copies of that big long chromosome. And then I need to make another one. So now I need to make a copy of the smaller one. So 5 beads on each side. 1, 2, 3, 4, 5. And then 1, 2, 3, 4, and 5. So my number 2 chromosome has gone through S phase, and now it has its duplicate. Now, something to remember is that when they go through S phase, the chromosomes itself are going to be joined at the centromere. And so this is your typical, when we think of chromosomes, usually you think about them like in that X configuration. That X configuration, like it's shown here on these 2 white ones, that X configuration is only true after S phase, only after the DNA has been replicated. Otherwise, the chromosomes don't look like those fancy little X's. Okay. Now I just need to do the same thing for my long number 1 that I got from my mom. So I'll connect 10 beads on one side. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 on that side. Kind of have to smash them together a little bit to get them to stay. And then 10 on the other side. 3, 4, 5. 6, 7, 8, 9, 10. Okay, so I just went through replication with my number 1 chromosome that I got from my mom. So it's crazy now, if you really think about it. On this 1 chromosome, I actually have 4 alleles present. I've got 2 alleles. They're going to be the same identical alleles, but 2 alleles of the same type on that one, 2 alleles of the same type on that one. All right, now I just need to replicate my number 2 chromosome I got from my mom. So 5 beads on each side. 1, 2, 3, 4, 5. All right. And boom, and then I just join them at the centromere, so the middle part is touching. The other parts don't have to touch. And then boom, there you have it. So we've just gone through S phase. Our DNA's been replicated. Because the DNA gets replicated in euchromatin phase, this is sort of misleading. The chromosome should be long and spindly and drawn out. Okay, so we repeated that process for each chromosome. We've now got each chromosome consists of 2 strands of double-stranded DNA. Oh, that's cool. You can just put it on the counter right there, bud. Thanks. And then you've got, these are the sister chromatids, and they're held together at a centromere. So you got sister chromatids joined together at a centromere. Now that the DNA has been replicated, we can simulate mitosis. So what I want you to do is take a picture of this. You need a picture of interphase, okay? And then we'll start our mitosis. So nuclear division starts with prophase. So there is no like, okay, phase 1 is done and it freezes, then it's phase 2. It's sort of like, it's a flow. It's a flow. And in that flow, we break down certain parts into phases. The first phase is prophase. And so once nuclear division is over, or once it begins, like once we start mitosis, several things are going to happen. The chromosomes are going to condense and become like they are now. They're going to become visible in the nucleus. The centrioles, that's these guys, are going to duplicate themselves and then migrate in pairs to opposite sides of the cell. And so I'm going to simulate that right now. There's my centrioles duplicating. And then they're going to move to opposite ends of the cell. Okay. All right. So we're going to, let's see. Where are we at? The mitotic spindle begins to form, nuclear membrane slowly dissolves. So we're going to do that. So we're going to remove the yarn that represents the nuclear membrane. We'll set that off to the side. And then we're going to separate the 4 chromosomes so they're not in a big jumbled pile, which is fine. Try to keep them together. Okay, so there we go. We got prophase. We placed, let's see, we added another pair of pennies, placed 2 pairs on opposite sides of the nucleus. There we go. And next, metaphase. All right, so what I want you to think about with metaphase is that we want to end up with 2 cells that have exactly the same things in them. So how is that going to work? Well, in metaphase, the chromosomes are going to line up on this center line that's going to divide them. And so what we want is we want one of these long white ones on one side, and we want one of these long white ones on the other side. And then that way there's 2 identical chromosomes, one in each of the new cells. So I'm going to kind of line that up on the top. I'm going to run out of room here, so I'm just kind of doing the best I can because my chromosomes are super long. And they're going to line up on this metaphase plate. Now when they line up, you know what? I'm actually going to turn it long ways and do it like this. Sorry, my chromosomes are falling apart. But this is going to be easier to see what I'm talking about. So here's the middle. I'm going to move my pennies off to the sides. What a mess. Let's see. 5, 6, 7, 8, 9, 10. And then these 2 came from this side. Okay. That's probably about as good as I'm going to get it without this whole thing just falling apart. Okay. It's hard for me because I got to make sure that everything's in view of the camera. Okay, so you can imagine these are all on one line here. And so what's going to happen in anaphase is that the sister chromatids are going to separate. And what you'll notice is that each side is going to end up with the same exact thing. They're going to be mirror images of each other. And that's different than what happens in meiosis. Okay? So here we go, metaphase. So they all line up on the center at right angles from the microtubules. Now microtubules are just long protein filaments that are going to extend from the centrioles, and they're going to attach to the centromere of the closest chromosome, sister chromatid, to them. So you got spindles are going to come out, these microtubules are going to attach to the kinetochore of the centriole. And then we begin anaphase. So 2 events. The centromeres, that's these long ones that have been holding the chromatids together, are going to split. So now each chromatid becomes its own chromosome. So when they're together, like in these pairs, they're called chromatids. When they separate, which is what we're going to do next, they are now chromosomes. And so they're going to separate, and they're going to start getting pulled by, now we didn't model the microtubules, but the microtubules are pulling on the centromere of each chromosome, and it's kind of creating some drag through this, like through the cytoplasm, and that's why they kind of V out behind them. So these sister chromatids are separating in anaphase, like so. So the sister chromatids separate. They separate from each other. And now you can see there's going to be one copy of the long chromosome I got from mom in one cell, one copy in the other cell. Same thing for the one I got from dad. I'm going to have one copy in one cell, one copy in the other. Same thing for the 2 short ones that I got from mom and dad. Okay. So this is anaphase. We separated the centromeres, and we pulled the chromosome to the vicinity of the centrioles. That's these guys right here. All right, so now we should have 2 identical piles of chromosome, one beside each centriole pair. Now you can move these a little bit closer if you'd like. Let's make piles. And that, my friends, if your chromosomes don't fall apart, hopefully they don't do this in real life. There you go. Okay. All right. And so now we can take a picture of this. So that's what I highly recommend, just taking a picture of it right now so you don't forget. And then we're going to go through telophase. And so telophase is the reverse of prophase, because now we have to uncoil the chromosomes, the microtubules have to go away, the nuclear membranes reappear. And so in telophase, we're going to, Avery, you can't be on my lap like this, bud, because you can sit in this chair right here and watch what I'm doing. But I got to be able to move my arms, bud. All right, so this nuclear membrane is going to show back up. For each one of these. And then I got another piece of yarn. Where'd my other long piece of yarn go? It's not this one. I'll just tie these together. Dun, 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 dun. And then you're going to take your other yarn and you're going to wrap it around the other pile of chromosomes. So, boop. And then telophase is done. Okay, so imagine that's wrapped all the way around. So I've now got 2 nuclei, each identical to each other in the same cell. Okay, so remember the purple string is just the nuclear membrane, it's not the cell membrane. Okay, let's see. Place circles of yarn around each chromosome. Oh, that's interesting. It did say to use threads to represent the microtubules. That's interesting, because I did not see threads in our list of what to have for this. So don't even worry about it, the thread. Just, this is fine. So we imagine the microtubules were in place. That will work. Okay, let's see. So this represents the 2 nuclei. And then now we have, after telophase, we have cytokinesis. And so this is where the cell membrane actually divides. And you can think about this like a ratchet strap. So you can imagine this is a giant bubble that surrounds these 2 nuclei. And then there's going to be a ratchet strap of proteins that go around the outside and then just slowly tightens up until the 2 bubbles pop, or until the 1 bubble pops into 2 bubbles, right? So that ratchet strap pop, and then now you have 2 bubbles, or 2 cells, each with their own nucleus, and each with identical sets of chromosomes. And so that's how mitosis works. That's how you get from 1 cell, that's how you get 2 identical cells. All right, and so in the next one, we're going to move on to, we are going to do meiosis. So meiosis, let's go ahead and reset. So I'm going to take off one set of chromosomes. We'll go back to where we were before when we started. So let's go meiosis. Dun, 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 dun. Just taking these off. I will use these later, so I don't want to disassemble them. Okay, and we'll go back to our regular old cell membrane. Imagine that these chromosomes in our interphase are pretty much, that looks like too many. One, that's 5, 10, 5, 10. I don't know where these came from. I guess they just got added to it. Okay, so now we have, this looks good, just like that's fine. Sorry, these clay beads like to fall apart pretty easily. Okay. All right, meiosis. So what's going to happen after meiosis, we're actually going to go through division, cell division, twice with this one. The purpose of meiosis, purely, think about meiosis this way. The purpose of meiosis is to create more genetic variability in the next generation. Why? Environments are always changing. Think about climate right now. Climate change is happening. So you've got environments are changing, ergo, the phenotype of the organism that exists today is based on the genotype. The genotype is what sets of alleles you have in your chromosomes. So your genotype determines your phenotype. Your phenotype is what interacts with the environment. If the environment is always changing, then you are going to want to also change the arrangement and the relationship of alleles on chromosomes to each other. Meaning, if you have, let's say on this long chromosome here, you have an allele, or let's say you have a gene that codes for fur, like thickness of fur. And on this one chromosome, you have an allele for that fur thickness that makes it really thick and really warm. And over here on this other chromosome, let's say that you have an allele for fur that's really thin, okay, and not very thick. And then on that same chromosome, maybe further down, maybe you've got an allele that has to do with water intake, like how much water you need to take in to survive. And so remember, this is the chromosome that has the thick fur allele. And then down here we have a gene for how much water you need. And on the same chromosome, you have an allele for water that says, you know what, you don't need a whole lot of water. You can survive with very little water. And over here on this chromosome, you have the same gene, but the allele says, no, you need to drink water constantly. So I have an allele up here for thin fur, and I have an allele down here for needing a lot of water. Up here I have an allele for thick fur, and an allele down here that says you don't need a lot of water. Now what can happen in meiosis, which is really cool, is we can take the allele for the thick fur and put it on the same chromosome as the allele that codes for needing a lot of water, and we can take the thin fur one, put it on the chromosome that has the allele for not needing a lot of water. And so what might happen is if you pass on the new chromosome that says, hey, you're going to have thin hair or thin fur, and you're not going to need a lot of water, now that chromosome in a new environment that maybe is dry, hotter, with less water, is now going to do better than if those alleles were back on the same chromosome. So that's just my spiel on meiosis. It creates genetic variability that can then match the changing environment. Okay. So anyways, the goal of meiosis is to end up with 4 daughter cells. We're going to divide twice. We're going to end up with 4 daughter cells, each with only one set of chromosomes. So, meaning one of those cells will have a long one and a short one. Every single one of those 4 cells will have a long chromosome and a short one. But what you'll see is that they will all be different. Okay, so let's carry on. Let's see, meiosis 1 and meiosis 2 are the 2 division cycles. We're going to get 4 daughter cells, each haploid, and they're all going to be unique, and you'll see why they're unique. And then ultimately those cells go on, depending on the gender, will become sperm or they'll become eggs. And so you would want haploid cells. You would want one set of chromosome in each cell. So if you have a sperm and an egg and they join together, now you have two sets, which is what we're used to. That's the diploid. Okay. All right, we're gonna take, do not forget this, we're gonna take a picture of prophase 1 and telophase 2 for submission. So I'll remind you when we get there. So we have interphase. Interphase happens just like it did in mitosis. Okay, so we're gonna just kind of put up our membrane. And the cell's just chilling, doing its thing. Maybe this is a germ cell that's in the gonads, so in the ovaries or in the testes. And it's just chilling, and then all of a sudden it receives a hormone signal. Hey, you need to go through meiosis. And so what this cell is going to do is this cell is going to need to duplicate its chromosomes, just like we did in mitosis. All right, so we're going to replicate each chromosome. I'm on step number 2 of interphase and meiosis 1. Step 2, as you did before, replicate each chromosome and attach the chromatids together. Okay, no problem. I already did that. Bloop. So there's one. So remember that this replication is DNA replication. This is, you know, there's going to be multiple origins of replication. There's going to be a replication fork. The polymerases are going to do their thing. And boom. And now we have replicated chromosomes. So we have 2 sister chromatids for each chromosome. We still call these a chromosome. It's kind of weird. We call that a chromosome, even though there's 2 sister chromatids, each with the same amount of information on them. It's just the nomenclature for this stuff is kind of funky. But with lots of practice, you'll get it too. Okay, so we just replicated our chromosomes. Meiosis 1, so we're going to have to go through 2 divisions. The first division cycle is divided into 4 phases, just like mitosis. There's a lot of differences, though, especially prophase 1, which is what we're about to go to. So please pay attention, this is important. Okay, we're going to separate the 4 chromosomes and place the homologous pairs side by side. So basically, we're going to take these guys, and we're going to put them side by side with their homologous pair, and I'm just going to do it like this, okay? So they line up next to each other in prophase of meiosis 1. Okay, so these guys line up next to each other, and they form a tetrad. All right, we're going to add centrioles to opposite ends of the nucleus. So I'm going to put those centrioles here. Actually, you know what? We'll do it the opposite way this time, because I don't have to line them up the same way until the second division. Okay. So boom, there we go. So I added the centrioles. I'm removing the yarn circle. And then the pairing up of homologous chromosomes is called synapsis. So they're entering synapsis. The non-sister chromatids, so that would be the white ones and the yellow ones from different colors, within the homologous pair are going to break and reattach to the other strand. This is called crossing over. So remember how I said, oh, you might have a gene here, like an allele that codes for thick fur or thin fur? Well, now I'm switching those alleles. So I'm going to take like 4 lumps of clay from this one. I'm going to put it on this one right there. And then same thing for this, so I'm going to reattach that there. So I got 4. Okay. Trying to figure out why it looks funky. It doesn't really matter that much. Okay, so we switched it there. And then maybe let's say like this end of this chromosome ends up switching with the end of this sister chromatid. Like that. And then maybe down here, we grab some beads. It does not matter which beads you switch, but just remember that what you're doing is you're putting alleles together of different genes that originated on different chromosomes. And so essentially what you're doing, what nature is doing, is nature is blind. She cannot see what she's doing. And so perhaps the outcome of blindly switching these around is something that's worse. It doesn't work as well. But occasionally you'll rearrange these chromosomes and you'll end up with one chromosome that's like a superhuman, or you know what I mean, like a super chromosome that has a whole bunch of great alleles on it, and the next generation that inherits that is going to be better off in terms of survival or reproduction. Now, same thing on this one. I'm just going to switch a couple, not that many. So we switch one allele over there. I'm going to switch. Ugh. Okay, looks like I popped 3 off. Let's pop off 3 of this one. Put those over there. Okay. These are all different enough now that when we go through our meiosis, we should end up with 4 unique cells. So that was enough crossing over. So that was crossing over. Notice, even after crossing over, I can still identify the original chromosome. I can say, oh, this white centromere indicates that was the one I got from dad, and that yellow one indicates that's the one I got from mom. What's interesting though is this one's still just yellow. Let's switch some of that guy around here. So I'm just gonna switch 2 of those with 2 of those. Boop, boop. There we go. Now they're all a little bit different. Okay, perfect. Moving on. All right, so we got crossing over. So cross the ends of 2 chromatids of a homologous pair. Oh, okay, whatever. We already did that part. We broke them and then we reattached them. Okay. Okay. So this exchange in pieces is a recombination of genes on DNA strands. This greatly increases genetic variability of the gametes produced, that would be sperm and egg, and consequently of the possible offspring. So this is what gives us variety. This is why your dad might have 2 different parents that are way different from each other. Maybe one of them has blue eyes, blonde hair, the other one has dark hair and dark eyes, and maybe your dad has dark hair, dark eyes. But because he has a chromosome from each one of those parents, a blonde hair, blue eyed, and a dark hair, dark eyed, is that now he can create a chromosome from crossing over that he passes on to you that has dark hair and light eyes, or blue eyes, or light hair and dark eyes. And so what you're able to do is recombine traits that normally wouldn't be passed on together. Okay, now make sure that you take a picture of this. So this is our completed prophase. We've passed everything, we've crossed things over. Take a picture of that, and we'll move on to metaphase. So as prophase 1 ends, the homologous pairs, the tetrads, are going to line up in the center of this cell. And so I'm making this the center of my cell. So notice how they're lining up now is different than how they lined up in mitosis. If I can get these without falling apart too bad. Now, how they line up is interesting. So let's see if it talks about it in the lab. Notice this alignment is different than how we did it in mitosis. Remember, in mitosis, we lined them all up and then split all the sister chromatids in half. Right now, we've got the tetrads lining up on the metaphase plate. The microtubules are going to attach. So again, we're using our imagination. Microtubules, little strings coming out and attaching to the centromeres of the sister chromatids. So the microtubules attach. When those events complete, the cell's in metaphase 1. All right, so we lined up the chromosome in homologous pairs, placed a length of thread between the centrioles and the centromeres. Ah, screw it, let's do it. Fine. I just have a bunch of strings, so I can handle this. In fact, let's do that. All right. There, boom, there's my microtubule. You happy? So remember, this is my filamentous protein. So this would be a structural protein that then is, this one also acts kind of like a ratchet strap. So there's 2 fibers, 2 protein fibers that are in each strand, and those 2 protein fibers use ATP and act like a ratchet strap, and so ATP gets burned up. Yes, the cellular battery gets burned up, and in the process of burning it up, combusting it, the 2 strands are like this, and so it shortens the lengths of string, which pulls the chromosomes apart. Okay, metaphase, so we got that. So now we're gonna do anaphase. The chromosomes move along the microtubules, so the microtubules shorten, homologous pairs separate to opposite sides of the cell. Now here's a part that's not mentioned in this lab, but I wanna bring your attention to it. Notice here that these 2 centromeres are yellow. That means they came from my mom. And over here, these 2 centromeres are white. Wait a second, so when these start to separate, which they're gonna do right now, when these start to separate and the microtubules shorten, that means, gosh, help me, help me. When these separate and the microtubules are shortening, goodness gracious, everything is falling apart. Okay, well, thank God our DNA is more resilient than my clay chromosomes. And kind of smash them together. There we go. Now you won't fall apart. Okay, so as these microtubules shorten and it pulls these homologous pairs apart, notice the homologous pairs stay together. Notice the sister chromatids stay together. So that one's getting pulled that way. That one's getting pulled that way. So this is anaphase, but notice that this is the part I want to show you. These homologous pairs of chromosomes aligned independent of one another on the metaphase plate. That adds another layer of genetic variability that is not discussed in the lab, but it's very important. Meaning, see how this one it has the 2 yellow centromeres and this one has the 2 white ones? Well, now we're splitting it up. Now I'm taking the 2 long yellow chromosomes I got from mom and the 2 short white ones I got from dad, and I'm putting them in the same cell. And notice I'm taking the 2 white ones I got from dad and the 2 yellow ones I got from mom, and I'm putting them in the same cell. On top of that, now I have also the variability of different chunks of or colors of clay on each sister chromatid. And so now that's adding to our genetic variability. It's independent, meaning that these 2 could be flipped. So I'd have 2 yellow ones on the right and 2 yellow ones on the right up here, and 2 white ones on the left and 2 white ones on the right. For every set of chromosomes. So for all 23 pairs, this can happen, flip-flop, back and forth. And so that's another reason you get genetic variability in meiosis. Okay, so anaphase. The chromosomes move along the microtubules, homologous pairs separate to opposite sides of the cell. Chromosomes still consist of 2 chromatids joined at a centromere. All right, separate each homologous pair and place the 2 chromosomes on opposite sides of the cell. Cool. So this one goes on this side. I'll try to smash these together a little better, because after this, they're not really being separated ever again. So I'm gonna smash, just gonna tidy this up a little bit. And I'm gonna smash these together a little better. Okay. And then same thing for these. I'm just gonna smash the sister chromatids together. I'm gonna smash these together. Okay. Now, boom. Those end up over there, those end up over there. Same thing, I'm gonna smash these together because they're not gonna separate again. We're not gonna have to switch alleles again. No. Okay. But you'll notice is that the centromeres are still joined together. So, boop, boop. the centromeres are still joined together. Okay, boom. So the centromeres are still attached, okay? Now, anaphase is over. Remember, telophase is just like that in mitosis. So the chromosomes uncoil, they become less visible, microtubules dissolve, nuclear membranes form. So we can actually, we put the two sets of chromosomes into quote-unquote disorganized piles. And I can get rid of this pile of beads and this pile of beads. Okay. All right, so there they are. There's my nuclear. This is such a shit show. Okay. There we go. And there we go. Okay. Bada boom. Now we go through cytokinesis. So cytokinesis splits the two cells. Now we got two cells. And notice that these two cells are diploid, but notice that the chromosomes they have are different from each other, meaning that the number 1 chromosome that we got from mom has portions on it that came from dad. They've been switched. And so same thing for the ones we got from dad down here, is they have portions of the chromosome from mom. All right, cytokinesis. Okay. Meiosis is not complete until the sister chromatids separate. So to achieve this, we got to go through nuclear division again. And then here we go. So that's all the new information, because meiosis 2 works just like mitosis. Meaning the sister chromatids are going to line up on the metaphase plate, and then they're going to separate, become their own cells, and then boom, Bob's your uncle, and you're done. So prophase 2, chromosomes condense again and become visible in the nucleus. So they go from euchromatin to heterochromatin. The centrioles duplicate themselves. So boop, you get one there, one there, one there, one there. The centrioles duplicate to opposite sides of the cell. Spindle apparatus forms, so string forms, and the nuclear membrane dissolves. So bloop, there goes our nuclear membrane. It dissolves. It dissolves. Opposite sides of the cell, opposite sides of the cell. And then we've got some short pieces of string to represent our microtubules. Metaphase. So what we're going to do in metaphase is the chromosomes are going to line up on the metaphase plate, just like this, just like this, just like this. So the chromosomes line up on the metaphase plate. So the metaphase plate is going to run like this. They're going to separate like this. Okay, and we've got our microtubules. We're going to send out our, probably cut these a little shorter now. Microtubules are going to come out from our centrioles and attach to the centromere of each pair. Those are microtubules. These are microtubules. Just need microtubule there. Looks like I need 2 more microtubules. Got them. And then this is another centromere, or another centriole. Centriole and centromeres are so confusing. They sound exactly the same. All right, and so this one is a little bit long, but it's attached. So these microtubules are connecting to the centromeres of each sister chromatid. That's metaphase. Okay, as soon as they attach, metaphase is over. Boom. Okay, metaphase 2, so they line up in a single file, center of the cell, thread goes to the centromere. Anaphase, so now these centromeres that have been holding the sister chromatids together split apart, and each chromatid now becomes its own chromosomes. The chromosomes will move along the microtubules, meaning these things will shorten, and these guys will split. So these guys shorten, Sister chromatids split. Hopefully our chromosomes stay mostly together. So here's our microtubules shortening, pulling our chromosomes apart from each other. Same thing with this one. So it's pulling the chromosomes apart, pulling the chromosomes apart. Sister chromatids separate, pulling the chromosomes apart. And there we go. So now what you'll see is look, there's a 1 chromosome, there's a 2 chromosome, a 1 chromosome, a 2 chromosome, a 1, a 2, a 1, a 2. Okay, so the chromosomes move towards the pole. There should be 2 identical piles of chromosomes, 1 next to each centriole. So I'm going to remove the microtubules, and I'm just going to place the chromosomes in a little pile next to each centriole. I could just finish pulling them. Okay. And this one as well. Just like that. Granted, it's all falling apart, but you guys get the idea. Okay, perfect. Okay, now moving on. So we're in telophase now, telophase 2, we're almost done. The chromosomes begin to uncoil, become nearly invisible, the microtubules are gone, we already took them away. The nuclear membranes appear, now you'll have a total of 4 nuclei. So I might have to cut some more string to represent this, but we'll do it. So there's 1 nuclear membrane forming around that guy. Looks like I'm gonna have to cut some more, huh, Eves? Yeah.